Microfluidics International Corporation leads the high-shear fluid processor industry. Microfluidizer processor technology provides consistent high shear rates with pressures up to 30,000 psi with the interaction chamber technology, ensuring seamless scale-up and unparalleled repeatability. These high shear rates enable the greatest levels of particle size reductions with uniform results, thereby offering proven success in key applications such as nanoemulsions, cell disruption, and liposomes. It is technology that's used extensively by leading companies in the pharmaceutical, biotech, chemical, food, and nutraceutical industries. This short video explains how the microfluidizer processor operates and why it is so successful. The process starts by first pouring the sample into the inlet reservoir. A constant high-pressure intensifier pump forces the liquid sample through the fixed geometry interaction chamber at very high pressures of up to 30,000 psi. Inside the interaction chamber, the sample is pushed through microchannels, which have been engineered to be typically 75 to 100 microns in nominal dimension, which is about the size of a human hair. When liquid streams are forced through these microchannels, it results in extremely high velocities of up to 500 meters per second. The stream accelerates and collides with both the chamber walls and itself, generating three types of forces: shear, impact, and energy dissipation. These forces disrupt cells and create the fine nanoemulsions that can easily be sterile filtered. Finally, the finished product is cooled in the heat exchanger or outlet cooling coil to control the temperature of the sample. The fixed geometry interaction chamber is the core of our technology and comes in different configurations to suit the application. The Y chamber is used for liquid-to-liquid dispersion applications, such as nanoemulsions, lipid nanoparticles, vaccine adjuvants, encapsulation, liposomes, and polymer particles. The Z chamber is recommended for suspension of a solid into a liquid in applications such as cell disruption, deagglomeration, and particle size reduction. With Y chamber, the two streams of liquid impinge upon each other to create the smaller droplet particle sizes, whereas with the Z chamber, the impact is up against the wall. Microfluidizer processors ensure that every milliliter of material gets the same high shear treatment, regardless of whether a 1 milliliter batch or thousands of liters per hour are being processed. By comparing the unprocessed to the processed samples, we can see just how effective the microfluidizer processor is. Control of particle size is achieved by adjusting the parameters of the interaction chamber type and size, the processing pressure, the number of passes, and the inlet and outlet temperatures. Microfluidizer technology is linearly scalable, which means that results achieved in the lab scale equipment is directly replicated in the pilot and production scale models. This scalability is due to the interaction chamber design. When scaling up, we simply place multiple microchannels in parallel to ensure every microliter of material is still being processed under the same high-velocity shear rate and impact forces. This ensures that the results are the same in the factory as they were in the lab. Many industry specialists have published papers indicating microfluidics as the optimal technique for the creation of nanoemulsions for drug delivery, vaccine adjuvant production, and cell disruption.